risk factors associated with it. It's telling you that it's involved. But first of all, so you know, ApoE does carry out the essential function. And it's functioning perfectly normally with respect to everything else. As far as I know, even if somebody is homozygous for the E4 alleles, they might have increased risk of cardiovascular disease. I'm not sure about that. I wouldn't be too surprised because it all has to do with handling of cholesterol. But the main genotypes with respect to ApoE have to do with Alzheimer's. Okay? So the liver has the highest, so it is functioning in other tissues. Brain has the second highest, and mainly it's astrocytes and microglia. Those are two real important cell types. Those are the only two cell types, but those are two of the three that the assigned paper focus on. Astrocytes, microglia, and neurons. Neurons, of course, and we'll see, are the ones that are transmitting the electrical, receiving and sending the electrical impulses. And by the way, there's one, those excitatory synapses, those measurements of the electrophysiological function. There's one figure in the assigned paper that has that. I have no way, I don't understand those experiments. Don't worry about that. Just take the word of the authors that there is a difference, okay? But this does carry lipids, okay? So once the lipids are bound with ApoE, it's called lipidation, okay? That's the transport mechanism for cholesterol being delivered to other cells. Cholesterol can either be biosynthesized inside the cell, or it can be transported from another source. And there's always a mechanism for very tight control of those levels, both through biosynthesis, uptake, and secretion, or not secretion, active transport, okay? Now this is important in the brain for synaptogenesis. Anybody know what synaptogenesis refers to and why it might be important for brain function?
Is it reading synapses?
Yeah, it's the synapse. So that's where one neuron, sends out those axons and dendrites, all those branches things. You've probably seen pictures of them, but they make connections in the brain with other nerves. And there can be those neurotransmitters being transmitted through the synapse, that space between the two basically processes from the neurons. In the neuromuscular system, there's synapses between the neurons and the muscles that are causing the muscles to contract. But this is all essential, so the brain cannot do without cholesterol. Okay. Now this is where I've added some slides from this extra supplemental review article, and it'll give you a lot more information about the normal functions of ApoE and also its connection to Alzheimer's. Okay. So all three isoforms are functional, so they differ in their handling of the lipids. Okay, but these, in ways that are still not completely understood, have to do, remember those plaques are formed because of abnormal aggregation of A beta and abnormal clearance of A beta. We'll see that it can actually be cleared. And all of this relates to, if you can read those words, synaptic plasticity and cognition, neurotoxicity, tau phosphorylation. Remember the tau phosphorylation that's responsible for those tangles made of tau. Neuroinflammation is a big deal. There's actually one of the cell types that is being looked at that's real critical to Alzheimer's. Basically access the macrophages are functionally equivalent to macrophages of the immune system functioning in the circulatory system and in other tissues. Okay, so those microglia cells are involved in inflammatory responses to damage, and they're contributing to the pathology with Alzheimer's. Okay, and just to give you an idea, I just took these. This is actually from a commercial site, but just to give you an idea, these are not to scale, so don't feel like these are humongous proteins compared to those. But this just gives you a little general idea about the different functions of these three main cell types. Thought to be important in the pathogenesis, but these are also the three cell types that are the focus of the assigned paper. and it's these microglia cells that I mentioned. They literally do present antigens just like macrophages. They are phagocytic, they will engulf and digest damaged cells, just like macrophages, cytotoxic, they will kill cells. So I don't think, this isn't the main mechanism for cell death. It's causing the symptoms in Alzheimer's, but obviously it's contributing to it. Okay, this is a figure out of the Tansey et al. I don't really like the figure, it's just too much going on. But once you get oriented to it, you'll see, I think you'll be able to see. So don't worry about all these other genes. These are just some of all the genes that are known to affect. But I want you to focus on where ApoE is. Okay. But there is a amyloid precursor protein. There is the positions of the cleavage sites in it. But this is where we're getting A beta 42 and A beta 40. Remember, there's something about that ratio of 42 to 40 that's significant. But what ApoE does is somehow it's contributing to that normal aggregation of these things, okay? And it depends on the allele of ApoE. ApoE4 is much more likely to cause abnormal aggregation, and ApoE2 actually protects against that, and again, in ways that aren't completely understood. But it also has to do with clearing these things from the brain, degrading. Now, ApoE isn't involved in the degrading, but you would want, normally you'd want this being degraded. That's what would happen normally. But when all of this aggregation is taking place, you're leading to plaques, you're leading to inflammation, you're leading to tangles, you're leading to neuron death of the neurons. Not something you want to do. But that's where it all ties in. Again, this is badly colored one, but I think this is a little bit more informative. This is from the extra review that I put up this morning. This is just about as far as I think you want me to read or look at in this supplemental reading. So here's our three main cell types. Astrocytes, microglia, remember these are the phagocytic ones that are acting basically at immune function. Here's the neurons that are dying off. And I added this little key. So ApoE, see how the ApoE is that protein? So that pinkish purplish is the protein. That carries lipid. It's just like HDL or LDL in terms of its function. But it's being expressed by either, see this arrow there and the arrow there? Both microglial cells and astrocytes express ApoE. But then there's cholesterol, there's lipid floating around outside of the cells, remember, okay? But then ApoE gets lipidated, those lipids attach to ApoE, and that's how ApoE is acting as a carrier for the lipids, for the cholesterol. And it carries it to the neurons. Neurons need cholesterol, right? Every cell needs cholesterol. You don't want it too high, but every cell needs it. And then it's taken up by LDL and R and LRP. LDLR is a receptor for the low density lipoprotein, okay? So like LDL, and then that's taken up by endocytosis into the cell. LRP is related to this, so both of these are the receptors for the lipidated ApoE. But it's also taken up, there's a feedback mechanisms, and this occurs even with liver cells and all cells, there's feedback. So, and remember, this is all part of the homeostasis of cholesterol. Some cells are biosynthesizing it, other cells don't have that ability, so other cells are dependent on the cholesterol lipids that are produced in one cell type. That first cell type that's synthesizing it exports it, carried through the bloodstream or through in the brain by ApoE and other protein carriers of these things, and then they're taken up by the cells that can't synthesize cholesterol. But if you have too much cholesterol, like in your bloodstream, it should be okay, because if too much cholesterol is going into the cells, it can also pump out cholesterol. But when there's defects in that pumping out, is where there's a problem. If you start getting accumulation of cholesterol inside the cells, it's bad. Okay, in terms of like atherosclerosis and things like that. Okay. Now this is what's important. Okay, A beta comes in here, where is it? I lost it. Okay, here's the soluble A beta. That's being produced from the neurons. Okay, so it's the neurons that are expressing ATP. It's the neurons that are having that processing by the secretase is taking place. Releasing that soluble, extracellular. Go back and look at the figures of the schematic. The soluble is being formed. That is being picked up, carried by the lipidase ApoE. Now this is where the isoform specific differences come in. The E4, because of those 2 amino acid differences, right? APO4 differs by 2 amino acids from APO2. APO3 differs by only 1 amino acid from each of the other 2. But they differ with respect to binding to A beta. Okay, and then that's where we're starting to get some problems. These are taken up and contributing to blood vessels. You know, the vasculature of the brain is really important. Okay, and then that's where things start going haywire in terms of these plaques forming. Okay, because of the abnormal aggregation of these plaques, because of ApoE is actually contributing to the plaque formation, because it's ApoE that's dealing with this soluble A beta, but in a way that gets locked up. Now, you do need to understand a little bit about intracellular. So when ApoE is taken up, okay, and where the APP is involved, okay, and the research paper refers to endocytosis and endosomes, okay, so you can refer to this paper. You don't have to, I don't think that you need to know anything else. It refers to early endosomes. I don't think it looks at late ones, but it refers to lysosomes. This is what's happening. This is the standard, this is the typical endocytosis pathway, happens normally. Okay. But what they find is that there's real early markers, or key markers of these early endosomes just getting packed full of A beta. That's probably contributing to the pathogenesis. This is a graphical abstract that was on the first page. This is actually really handy. They made iPSC cells. Now, I was a little disappointed. They only did the first set of experiments were done with one person, iPSC cells from one person, which, a sample of one, even when they're making isogenic strains, right? So they converted the healthy iPSC. They didn't say whether the donor was homozygous or heterozygous for ApoE3, but regardless, through CRISPR, they changed it. It's a missense substitution difference. They changed it to ApoE4. Then they differentiated into the 3 different cell types. Then they'd have isogenic, differentiated, normal, well, I wouldn't say normal, that have their homozygous for ApoE3, and normal, but that are homozygous for ApoE4. Not even having any Alzheimer's in here. They're not, I don't think they even refer to if they sequence the 3 alleles, no genes, no involved. I'm pretty sure there's no reason to think that there would be anything wrong with these other than being different with respect to the ApoE isoforms. And this is just summarizing for you what they looked at. Okay, you can look at them. I think that there's online. There's a video abstract. A lot of journals are doing this. This is in the journal of Neuron. This is the scientists, 3 of the scientists that were involved in this study. I downloaded the video just in case you're off campus and try to get it. You might not be able to get access to it off campus. But I'll upload it into ELC so you can look at it on your own.
It is well known that carrying the ApoE4 allele conveys a higher risk for Alzheimer's disease than carrying ApoE3. About 30% of the population carries ApoE4. But there are important gaps in what the field knows about how APOE4 can affect the brain.
In our new research in neurons, we sought to understand the different APOE4 makes that may lead to neurodegeneration. We went under the hood to better understand what is going on in human cells and the models. We created tissue cultures and the 3D organoids from human-induced pluripotent stem cell, or iPSCs. The organoids are not really brain, of course, but they are models where T cell works in similar ways. We use CRISPR-Cas9 gene editing to insert the APOE4 T variant into iPSCs derived from healthy individuals with the APOE3 phenotype. We also compare this to isogenic APOE3 controls.
It was a huge effort in my lab that has produced an important set of results. With the finding of this detailed new study, we have a much clearer picture of how APOE4 leaves the brain vulnerable to neurodegeneration.
For us, getting under the hood meant looking at the key cell types in Alzheimer's disease neurodegeneration. We looked at neurons, astrocytes, and microglia-like cells in our 2D cultures, as well as at cerebral organoids. In our APOE3 and APOE4 models, we were able to see significant changes in cell function related to amyloid and tau. We started out by using RNA sequencing to track gene expression differences. We saw hundreds of differentially expressed genes in neurons and even more in astrocytes and microglia.